For decades, the only way to check an unborn baby's chromosomes involved a needle pass through the mother's abdomen and into the womb itself, a real, if small, risk to the pregnancy. Today, that same information often comes from a simple blood draw. This is how that became possible. The test goes by a few names: NIPT, non-invasive prenatal testing, or cell-free DNA screening. And all of them describe the same idea: reading fragments of the baby's own DNA that are already floating in the mother's blood without ever touching the baby directly. Reframe what that actually means for a moment. Somewhere in a routine tube of blood drawn from a pregnant woman's arm, genuine fragments of her baby's DNA are already present. Has been the whole time, just too small and scattered to notice until scientists knew exactly what to look for. The scale of adoption has been enormous. This test is now offered to millions of pregnant women every year worldwide, and in many countries it's become a routine part of standard prenatal care rather than a specialized, hard-to-access option. At a glance, here's what makes it different from what came before. No needle near the baby. Results in about a week. Available from roughly 10 weeks into pregnancy, and meaningfully better accuracy than the blood and ultrasound screening methods it's replacing. The basic process is simpler to describe than you'd expect. Blood is drawn from the mother. The plasma is separated out. The DNA fragments floating in that plasma are sequenced, and a computer compares how much DNA came from each chromosome. Here's a genuinely fascinating fact to sit with for the rest of this video. The baby's DNA isn't coming from baby cells at all. It's coming from the placenta, an organ the baby builds, but one that's technically separate tissue, constantly shedding its own DNA into mom's blood. Keep that placental detail in mind. It's going to matter a lot later in this video, both for how the test works and for the specific ways it can occasionally get things wrong. This entire technology traces back to one researcher's unusual insight in 1997, and that insight didn't come from studying pregnancy at all. It came from studying cancer. Dennis Lowe, a Hong Kong-based physician scientist, had read that dying tumor cells release fragments of their own DNA into a cancer patient's bloodstream. He made a genuinely creative leap. A growing pregnancy, he reasoned, resembles a large, rapidly growing tissue mass too, so maybe it releases DNA into the blood the same way. To test the idea, he needed a way to tell fetal DNA apart from the mother's own. So he focused specifically on pregnancies carrying a male fetus, searching maternal blood for Y chromosome DNA that could only have come from the baby, since the mother herself has no Y chromosome at all. The result confirmed his hunch. Depending on the exact sample type used, fetal DNA was detectable in 70 to 80% of the pregnant women tested. Clear, genuine proof that a baby's DNA really does circulate freely in its mother's blood. As you might expect by now, the finding was met with real skepticism at first. It took years of further work before the scientific and medical community accepted that this circulating DNA was reliable, abundant, and clean enough to actually build a clinical test around. From that first 1997 paper, it took roughly 14 years before the first commercial clinical test reached patients in 2011, and just over a decade after that, it had become routine prenatal care across much of the world. A single creative analogy: a pregnancy is a bit like a tumor, biologically speaking, quietly launched one of the biggest changes in prenatal medicine in a generation. Now let's slow down and actually understand the biology behind this, because the answer to what exactly is floating in that blood sample is more specific and more interesting than most people assume. Every one of us, pregnant or not, has small fragments of DNA circulating freely in our blood plasma, released whenever cells die and break apart in the ordinary course of the body renewing itself. Scientists call this cell-free DNA. This is that same routine procedure, a standard blood draw from the arm, no different from any other blood test, that carries all the information this entire technology is built on. Trace the actual chain of events. Placental cells die naturally as part of normal turnover. They break apart and release their DNA. That DNA enters the mother's bloodstream, and a blood draw captures a snapshot of it. During pregnancy, a second source of cell-free DNA enters the picture, the placenta. Placental cells naturally die and renew constantly throughout pregnancy, releasing their DNA into the mother's bloodstream. And because the placenta develops from the same fertilized egg as the baby, its DNA is essentially identical to the baby's own. The proportion of placental DNA in that mixed sample is called the fetal fraction, typically around 10 to 15% of all the cell-free DNA in maternal blood by 10 weeks of pregnancy, with the rest coming from the mother's own cells. That fraction has to clear a minimum threshold, usually around 4%, for the test to work reliably, and several factors can push it below that line. Testing too early in pregnancy, a higher maternal body weight, or certain twin pregnancies. That threshold is exactly why the test isn't usually offered before roughly 10 weeks. Earlier than that, there simply isn't reliably enough placental DNA in the sample yet to get a trustworthy result. So the whole test rests on a beautifully simple biological accident. The placenta constantly sheds DNA, nearly identical to the baby's, into a place doctors can easily reach with a syringe. With placental DNA sitting in that blood sample, the actual analysis comes down to a clever statistical trick, one that doesn't require reading the DNA's meaning at all, just carefully counting it. The lab uses a technique called massively parallel sequencing. Millions of tiny DNA fragments in the sample are read simultaneously, and each fragment is matched back to exactly which chromosome it came from. If a baby has the usual 2 copies of chromosome 21, fragments from that chromosome should make up a predictable, consistent share of the total. If the baby has 3 copies instead of 2, that share statistically creeps up, a small but detectable shift called a z-score, worked out with real numbers. If chromosome 21 fragments normally make up about 1.3% of all sequenced DNA, a trisomy 21 pregnancy might push that to roughly 1.36%, a shift of only a few tenths of a percent, but statistically unmistakable across millions of counted fragments. Visualized on a gauge, a z-score comfortably crossing the flag threshold is exactly what tells the lab software to report the pregnancy as high risk for trisomy 21, a purely statistical call, made from proportions, not from reading a single gene's actual function. Compare that directly to what came before it. Older combined first trimester screening, using ultrasound measurements and blood hormone levels, caught roughly 85 to 90% of trisomy 21 cases. Cell-free DNA screening catches over 99%, with a dramatically lower false alarm rate. Here's a fact worth sitting with. This test never actually reads what any single gene does. It simply counts how much DNA came from each chromosome and compares the proportions. Remarkably simple math producing remarkably precise medical information. Counting, not reading. That's the entire statistical trick behind one of the most accurate screening tests in modern medicine. Understanding the statistics is only half the picture. Just as important is understanding exactly what this test is designed to catch, and, critically, what it is not designed to do. Quick pause. If this breakdown is helping you, take a second to hit like and subscribe. It genuinely takes a lot of effort to make videos like this, and your support keeps them coming. This is a real karyotype showing trisomy 21. 3 copies of chromosome 21 instead of the usual 2, the chromosomal basis of Down syndrome, and the single most common condition this screening test is designed to detect. The core conditions this test screens for are the 3 viable trisomies, extra copies of chromosome 21, 18, or 13, along with abnormalities involving the sex chromosomes, and it can also reveal the baby's sex as a byproduct of that same analysis. Here's the single most important distinction in this entire video. This is a screening test, not a diagnostic one. A screening test estimates risk with high accuracy. A diagnostic test, like amniocentesis, directly examines the baby's own chromosomes and gives a definitive answer. Please take this seriously. This video is for general education only. It is not medical advice, and a positive or high-risk NIPT result is never a final diagnosis on its own. Any result, especially a positive one, should always be discussed with your doctor or a genetic counselor and confirmed with diagnostic testing before any decisions are made. A dangerous myth worth killing directly. A positive result means the baby definitely has the condition. It doesn't. It means the risk is high enough to warrant a diagnostic test. The actual presence of the condition still needs to be confirmed. For trisomy 21 specifically, the numbers are genuinely excellent. Sensitivity above 99%, meaning it catches the overwhelming majority of true cases, and specificity above 99.9%, meaning it very rarely flags a healthy pregnancy incorrectly, framed the way a lab would frame it. This is formally a screening test with published sensitivity and specificity figures, and its real-world predictive value shifts depending on how common the condition is in the population being tested. Line the two test types up side by side, and the relationship becomes clear. Screening comes first, is low risk, and estimates probability. Diagnostic testing comes second, carries a small procedural risk, and gives a definitive answer. Extraordinary accuracy is still not the same thing as certainty, and understanding that difference is exactly what this technology asks of the people who use it. Remember that detail from earlier, that the DNA being tested comes from the placenta, not the baby's own cells directly? That single fact explains nearly every strange way this test can occasionally give a misleading result. Occasionally, a chromosomal abnormality exists only in placental cells, while the baby's own cells are completely normal, a phenomenon called confined placental mosaicism. The test reads the placenta correctly, but the placenta simply isn't telling the whole story about the baby. Another genuinely strange source of false positives: a vanishing twin. In early pregnancy, a twin sometimes stops developing and is naturally reabsorbed, but that twin's placental DNA can linger in the mother's blood for weeks, occasionally skewing the test's chromosome count. The rarest and most striking cause. In exceptionally uncommon cases, an unusual result has led to the accidental discovery of an undiagnosed maternal cancer, because the mother's own tumor was quietly shedding DNA into the exact same blood sample. There's also a subtler statistical reason results can be misread. The chance a positive result is truly correct depends partly on how common the condition already is in that pregnancy. And trisomy 21 becomes meaningfully more common as maternal age increases. That's why the same positive result can carry a different real-world meaning for a 25-year-old versus a 42-year-old. The test's raw accuracy doesn't change, but the underlying odds it's confirming genuinely do. None of this makes the test unreliable. It makes it exactly what it claims to be: an extremely accurate statistical estimate, built on real biology that occasionally has its own quirks. Remember the insight that started this whole story, that a pregnancy sheds DNA into blood the way a tumor does? Dennis Lowe didn't stop at pregnancy. He turned the same idea directly back toward cancer itself. Just like a placenta, a tumor sheds its own DNA into the bloodstream too, called circulating tumor DNA, or ctDNA. Sequencing it can reveal genetic mutations specific to cancer, sometimes before the tumor is even large enough to see on a scan. Set the two applications side by side, and the parallel is striking. In pregnancy, the placenta sheds DNA revealing a baby's chromosomes. In cancer, a tumor sheds DNA revealing its own genetic mutations. Same blood draw, same sequencing principle, entirely different target. Dennis Lowe's own later research applied this directly to nasopharyngeal cancer, a throat cancer strongly linked to Epstein-Barr virus and unusually common in southern China, using viral DNA fragments in blood as an early warning signal, the same way Y chromosome DNA once flagged a male fetus. The applications keep expanding beyond that too, monitoring whether a cancer treatment is working by tracking ctDNA levels over time, and even checking for early rejection in organ transplant patients, using donor-derived cell-free DNA in the recipient's blood. Grading the evidence honestly, prenatal cell-free DNA screening is now extremely well established with over a decade of validation. Cancer liquid biopsy is younger and rapidly evolving, genuinely promising, with a growing but still maturing body of clinical evidence behind it. A single insight about pregnancy and cancer sharing the same underlying biology has quietly become one of the most versatile tools in modern medicine. Powerful genetic information delivered this easily, this early in pregnancy, doesn't just raise scientific questions, it raises real, human ones too, worth naming honestly before we close. Access has genuinely improved over time. The test cost has dropped substantially since 2011, and it's increasingly covered by insurance or national health systems in many countries, though real cost and access gaps still exist worldwide. The ethical questions are genuinely worth sitting with, not glossing over. Earlier, easier genetic information changes decisions parents face. Some countries restrict revealing fetal sex specifically because of sex selection concerns, and equitable access remains an unresolved global challenge. One more time. Clearly, This video is general education, not medical advice, and cannot replace an actual conversation with your doctor or a genetic counselor. Any decision about whether to test and what to do with a result belongs with qualified professionals and the people actually living that pregnancy. If you're actually considering this test, a genetic counselor can walk through the real questions worth asking beforehand. What exactly will be screened, what a positive result would mean for you, and what your actual next steps would look like either way. Quick check before we close. If a screening result comes back positive for a trisomy, what should happen next? According to everything we've covered, from a cancer research insight in the 1990s to a routine part of prenatal care today, and now reaching toward cancer detection itself, this entire field is still remarkably young and still actively being written. A blood draw that once could only tell you a few basic things about a pregnancy can now responsibly, accurately screen for chromosomal conditions and quietly reveal a whole new way to catch cancer early, all from one small, elegant idea about DNA floating where nobody thought to look. Here are the key references and sources for this video. Thanks so much for watching. If this helped, like the video and subscribe for more clear, simple explainers, and I'll see you in the next one.